Is now starting. All attendees are in listen-only mode.
Hello, everyone. My name is Sherry Lynn Phelps. I am the Agronomy Manager with Saskatchewan Pulse Growers and will be moderating this webinar today. We also have Andrea Lauder, our Communications Manager, and our newest staff member, Jasmine Hansen, who is our new Marketing and Communications Coordinator with us as well. We all welcome you to this October 21st webinar. Bit of an on the administration side before we get started. To get CCA credits for today's webinar, you must be watching it live. An email will be sent out after the webinar requesting CCA numbers. If more than one person is watching, you will need someone to verify those in attendance and can include all the CCA numbers. The webinar will be recorded and posted to our saspulse.com website for future viewing for those unable to attend or those who want to have another look. A link will be provided to you once this is available. We also have a new fact sheet that pertains to this topic, as well as a podcast that will be done with Dr. Chatterton, and both of those will also be available very soon. All links to this content will be sent out into an email to you guys that have registered. For today's webinar, all participants will be muted. We will be happy to take questions at any time during the presentation. To ask a question, please type it into the question box located in the GoToWebinar dashboard. So that's on the right-hand side of the screen. For today's webinar, we hold all questions to the end of the presentation where they will be addressed. Today's speaker is Dr. Syama Chatterton. She is the Pulse and Special Crops Pathologist with Agriculture and Agri-Food Canada out of Lethbridge. Her research focuses on management of root and foliar diseases of pulse crops and molecular diagnostics and identification of soil-borne pathogens. She currently leads numerous research projects on pea root rot, white molded dry bean, and chocolate spot of faba bean. Besides her passion for pulse diseases, she also has a passion for cooking with pulses, particularly trying all sorts of different tricks to get her kids to eat pulses. She will be providing an update on her current research on Aphanomyces root rot, starting with a summary of results of the 4 years of work from the surveys in Alberta and Saskatchewan. She then will provide an overview of the lab research determining threshold concentrations of Aphanomyces that cause the disease, as well as the field distribution. Finally, she will discuss challenges and lessons learned from the field aimed at evaluating management options and predicting risk of root rot for 2021. I will now turn the presentation over to Dr. Chatterton for the Pulse Agronomy webinar titled Root Rots, Predicting Risk into 2021.
Hey, thank you, Sherry Lynn. I'm happy to be back here, I think about 6 months later from the last SPG webinar. And at that time, I think we were all just going into lockdown, and I thought things would look a lot different here about 6 months later, but I'm still working from home. And so today I'm going to talk about kind of what happened with root rots in 2020, and then how we can predict risks into 2021. And an overview of my slide here of the presentation. So I'm going to start with a pretty short overview of Aphanomyces euteiches. I think at this point, we're all very familiar with this pathogen and this disease. So I don't want to spend too much time on that, but we'll just give some quick reminders about the pathogen. And then I'm going to talk about 2020 and how we can use a 2020 growing season to set a baseline for into the future for planning for peas and lentils. And then I will spend the majority of the talk talking about reducing your risk of infection in 2021 and what we can start doing to make decisions now for the pea and lentil crops in 2021. Sherilyn mentioned that there's some new resources up on the SaskPulse website. And really, I've structured my talk around this new fact sheet that SaskPulse growers have just put out, talking about reducing root rot risk for 2021. And so this is to provide a little more detail on that fact sheet. And I also can't cover everything that is in that fact sheet. And I thought a long time about whether I wanted to begin my presentation with this kind of risk analysis guide that SaskPulse has put together, or end my presentation with this. And I decided to start with it just as kind of to show it as a roadmap of where the presentation is going to be going. So Sherilyn, I think, spent a lot of time, whoops, go back. We spend a lot of time discussing what to put together for kind of major influencing factors for Aphanomyces, some of the intermediate factors, and then the minor influencing factors. At first, my intention was to talk about all of these, but as I started putting the presentation together, and I think I'm already up to about 51 slides, I realized that there was no way that could happen within about 45 minutes. So I'm going to spend most of my time talking about these major influencing factors and then touching on some of the intermediate factors. But you can take a look at this kind of risk analysis worksheet, again, on the SaskPulse website. So just a quick overview of Aphanomyces. Its disease cycle is very important to managing the disease, or particularly why it's so difficult to manage the disease. And really the number one reason is because of these little beasts here, oospores. These, I think everyone should be familiar with these by now. These are these very thick-walled, you can see that very thick wall here, double cell wall, very thick-walled resting spores that basically will sit in the soil until it has the ingredients it needs to cause disease. And those two ingredients are host roots and water. So it'll sit there until you either plant pea or lentils, you get the rainfall into May and June, and these guys start to hatch, and what they hatch is to produce these zoospores. They get kind of released into these structures, so the zoospores all cluster at the top, and then the zoospores get released, and you can see them in this video starting to swim around, and you can see that they are swimming towards, these are pea roots here. You can see them start to swim towards there. And then once they find a pea root, they very rapidly kind of attach and insist themselves on those pea roots, and then they start to produce these infection structures called hyphae, kind of these strands of the pathogen, and it quickly can ramify throughout the whole roots. And really what it does, it just eats away at the outer layer of the roots or the root cortex until it gets pretty much decayed away. And then when there's no food left for the pathogen, it goes back to making these oospores, or these thick-walled resting spores. And so just some key aspects of the disease cycle. As I mentioned, oospores require the presence of a host to break dormancy, and that's really where a lot of the challenges come in with management. Nothing does live forever, though, and so they do naturally decay in the soil. And some research about 25 years old now kind of theorized that they have a half-life of approximately a year. And we'll look at what that means later on in the presentation. Under dry conditions, the oospores can infect the roots directly, but it's not the most efficient method of infection. So you'll see a lot lower infection if it's just oospores infecting. Again, one of the big challenges for this disease is that there's a very low threshold of inoculum for disease to develop, and that's about 100 oospores per gram of soil. And I'll talk a lot about what that means later on in the presentation. And so then in terms of zoospores, the zoospores on the multiplier stage. And so even though the oospores are what are sitting there in the soil, it's the fact that they can kind of multiply up, 100-fold to produce these zoospores that then infect the roots. And the zoospores do require saturated or wet soils to swim to find the roots. And that's why we see that the disease is much worse under wet conditions. Lentils are just as susceptible as pea for the most part. We'll talk a little bit about when that sometimes changes. And because of this fact that the oospores sit in the soil for so long, it's really the soil moisture and the crop history that are drivers of the disease. And I'm going to talk a lot about that throughout the presentation. So we're going to start with 2020 and setting a baseline. And really why this is important is because what happened this year in your pea and lentil crops has consequences for down the road. And unfortunately, 2020 did end up being a fairly, I'd say, good moisture conditions for Aphanomyces. That means poor moisture conditions for us that has to deal with this disease. And what we can see in these precipitation maps from Ag Canada is that this is the past 30 days of the June 30th. So June was a fairly wet month across all the prairies, except for maybe down into southern Saskatchewan. We had pockets of very, very high moisture in Alberta, and lots of rainfall as well in Saskatchewan, and then a lot of rainfall in Manitoba. And then things got a little bit better into July, but again, we see these really, really high pockets of moisture kind of in the northwest growing region of Saskatchewan, and it stayed very wet as well in central Alberta. And so this really created ideal conditions for Aphanomyces. And we all know that 2020 was a very unusual year. And so we didn't actually get to go out and do surveys, partly because of the COVID restrictions on what we were allowed to do over the summer. So I don't have kind of hard numbers on what we saw. I do have some pictures that I took while I was traveling to hikes or on vacation, where that's kind of how I did my very informal surveys this year. And this was even down in our dry area of Lethbridge that actually ended up being quite dry. This is mid-July, and we can see that the root rots really were showing up. This is right at the field entrance, and we can see a lot of damage, and then these widespread kind of yellowing patches throughout the field. And here's another one again near Lethbridge, which, as I said, ended up being fairly dry, and we can see kind of extensive Aphanomyces. So from what I gathered from talking to people, from some of the phone calls that I fielded, actually from looking a lot on Twitter, Aphanomyces was a really rough year for Aphanomyces, and I think that's partly why we're doing this webinar today. I did get some hot off the press data from one of my colleagues here from Alberta Agriculture that did some surveys, and he tells me that for Alberta, we did see about 100% prevalence of root rot. So that means all fields that they surveyed had root rot, with about a 67% incidence. So when we talk about incidence, we're talking about going into these fields and stopping at 10 spots per field. And that means a 67% incidence would mean that on average, 6.7 out of the 10 spots in every field in Alberta showed some root rot. Lentils, again, fared a little bit better in Alberta. We saw 90% prevalence, but only 14% incidence. And that's often what we see for lentils, is that some of the lentil fields, we don't see it as widespread throughout. So what can you do now that the growing season is over? And I would say the most important thing, and really I think kind of the take-home message and what we really want to start preaching about this disease, is that it is very, very important to start recording how your pea and lentil crop performed every time it's grown. Because as I said, crop history is one of the main drivers of this disease. So if you start to take note of how your pea and lentil fields performed, take note of where some of those small spots might be, that's going to be our best bet for making decisions going forward. And so how your pea and lentil crop performed in 2020 is going to determine the crop interval or the rotation interval that you're going to need, and will affect decisions for that field in 4 to 8 years down the road. Now, I've been very specific about saying 4 years here, because that's another thing that we really want to start preaching, is the importance of a good interval between pea and lentils. So some of the things that you can either reflect on now, or maybe you already collected some of this information during the growing season, is how your pea and lentil fields looked. Did you notice small yellowing patches in those fields? Was there yellowing along water tracks or on seepage areas off of side hill seepage areas from hills? Was there some spots that maybe yielded a little bit poorly than the rest of the field? And those are the kind of the information that we want to start paying attention to. Did you have full yellowing? Because that has a lot of implications. And then something that I really want to start driving forward as well as a take-home message is to check your roots during the growing season. And kind of a spoiler alert, I'm going to end my presentation with a reminder on checking your roots during the growing season. So I put together kind of a few pictures of what your pea or lentil, these are all pea fields, what your pea fields may have looked like in the summer. If you were fortunate, we'd say congratulations if you had a pea field that looked like this, then you're okay to plant pea or lentil in 2024. Again, I'm saying 2024 because we want to make sure that we have that kind of 4-year rotation between pea and lentil crops to maintain the stability of the soil. I'm going to say, there's some, as I go through the rest of the presentation, there's other factors that you want to take into account, but for the most part, if your pea field looked really good, then you're probably on track for having a good crop into 2024. If your pea field looked like this, or lentil field, then this is a field that you have to take out of pea or lentil production for at least 6 to 8 years, maybe longer. And we're going to talk about some of those, what some of those intervals should look like. If you had fields like this, where it's actually a very, very healthy pea field, but then you start to see these yellowing spots in the low spot of the field, we can see this is where all the water collects. You can see this yellowing along the water tracks. Or here another one, nice pea field, but you're starting to see these yellowing along the tracks. These are the fields that are a little more difficult to decide what to do with them. And these are the fields that is very important that you start doing soil tests. And I'm going to talk a lot of the next few slides about doing fall soil testing, while I guess how your pea field looked and or pea or lentil field looked is still fresh in your mind. Having said that, we're under a snowfall warning here in Lethbridge. I think it's supposed to snow 50 to 20 centimeters. Maybe we're already too late for fall soil testing. We'll have to see how the rest of the fall plays out here. But it's important to fall soil test, like I said, while this is fresh in your mind, so you can go to these spots where you know that there was some yellowing, where you know that there's maybe some yield loss, and collect from those. So I'm going to talk about post-harvest fall soil testing. But all the soil testing methods and the mechanisms for soil testing will be the same if you're going to do a pre-plant spring soil testing. And what I want to do is take out, I guess, some of the unknowns, or give you some information on how to soil test, what are the advantages and disadvantages, and how to use this knowledge. So I'm going to talk first about soil testing methods. This is my crew soil testing in a pea crop last year in 2019, when we were allowed to go into fields. And you can see that we use two different methods to collect soil, and either are fine. You can use a Dutch auger and kind of take some samples all throughout the field, or you can take a shovel and just take bulk soil samples again all throughout the field. We always wear booties in the field because we don't want to spread Aphanomyces from field to field. So this idea of being phytosanitary or trying to minimize the spread of Aphanomyces between fields is also very important. And we also always sterilize our equipment, usually just with bleach, as we move from field to field. So there's two soil testing methods that are most widely in use. The first is a soil bait test. And on this one, we usually collect a fair amount of soil, usually about a kg or so of soil from the field. Take about 500 grams. And just pot it up into any type of pot, it doesn't matter. We started out with fairly large pots, and now we moved down to about 2-inch pots, because you don't need that much soil to grow peas. And the nice thing about this is that you're taking this bulk soil and basically just transferring it straight there. So you get all that complement. These are my little oospores here. And then we plant pea seeds. We do treat the seeds with metalaxyl and fludioxonil so that we eliminate Pythium and Fusarium as potential contaminants. And then you just grow these for 3 to 4 weeks. And then you can see that you can get symptoms of Aphanomyces, really any root rot pathogens, quite nicely. We can see differences here between kind of different spots in the field. We dump out the soil, wash the roots, and then for the most part, the symptoms on the roots can be fairly obvious, so we can tell right away that this is Aphanomyces. Because we're researchers and we tend to have to back everything up at least 3 times, we do follow up with 3 different ways. One is checking for presence of oospores, and the second is plating them out to make sure it's Aphanomyces, and then we also do a DNA test. But what I wanted to say about these soil bait tests is that they're actually quite easy. You could even do them at home on your windowsill, as long as you can pot up some soil into some small pots and plant some peas. You can do your own soil bait test. Of course, you won't be able to do the follow-up, but this is one of the easier methods. It's just very labor intensive. The other method that we use is DNA extraction followed up by quantitative PCR. So I'll just run through those steps. So again, if we take our 500 grams of soil, we have our oospores in there. We take a small aliquot, 250, usually to up to 400 milligrams of soil. And these get put into these special tubes that come filled with a buffer and some beads. And these get ground at fairly high velocity. And really what we're trying to do is crack open all those cells to release the DNA. This kind of gets moved over. We do a whole bunch of series of washing and adding of buffers. And what we're trying to do here is remove all the inhibitors and just stabilize the DNA. And we'll end up with pulling out about 100 microliters of DNA extract. And then we take that, and we take about 2 microliters of DNA extract, put it in through this quantitative PCR. It's basically just a series of amplification steps so that we're ramping up the numbers of our target DNA in the tube. And then we can measure how much is left, or how much results in that tube. And then the idea is whatever our measurement here, we can relate back to what that number of oospores are in the soil. So that is theoretically how it's supposed to work. I'm going to talk probably a few slides down the road. We're going to talk about what some of the challenges are with this method. So just keep that in mind. And then what I've tried to do here is just list what the advantages and disadvantages are of each method, so that when you're going in and testing your soil, you have some knowledge on how this is supposed to work and can make the decision on what type of test you want to do. So one method is culturing that I didn't show an example of, and that's because it's actually impossible to do from soil. Those oospores are just tricky little beasts, and for whatever reason, they will not germinate in culture. They need that host plant to germinate. But it can work from roots, and one of the advantages is that it's very fast and it's fairly cost efficient. There is some tricks in being able to identify Aphanomyces in culture from roots, just because it can be easily confused with others. So you do need some expertise there. The soil bait method that I went through, it is what we're finding to be the most reliable method. And this is because it uses a representative size soil sample. Even though it's only about 500 grams, it's more representative for your field. And it can be semi-quantitative in that you can kind of determine the number of plants that show infection is related to how many oospores you started with. The big problem with the soil bait is that it takes a long time. It takes 3 to 4 weeks. It's more labor intensive because you have to pot up the soil, wash the roots. And sometimes the root systems can be confused for other pathogens, so you might need to follow up with culturing or with the DNA extraction and qPCR. And so in terms of this last method, it's definitely the fastest. You can get results within about 4 hours. It's very accurate because it's only going to detect Aphanomyces in that sample. It's not going to, you're not going to be confused with other pathogens or anything else. And it's also very quantitative. The biggest problem is that it's not always reliable, and the false negative rate can be high. And I'm going to go through that in about the next 5 to 10 minutes as we move into what you can do in 2021. So just kind of keep that in mind, and I'll tell you why a little bit later on. So for soil sampling methods, what we recommend is to remove that stubble layer. Because Aphanomyces doesn't move up from the root to the stem, you're really not going to find much in the stem. So you want to remove all the stubble layer, and it's not going to be present in previous crop stubble because it doesn't grow there. So remove the stubble layer, and then take the soil from the top 15 centimeters within a row. And this is particularly important for fall, post-harvest fall soil sampling, because you want to make sure you have some of those decayed roots in the soil sample to kind of reduce the probability of having a false negative result. Sample from low spots in the field or areas that have history of yellowing or poor yield. And this is where I said it's important to kind of record or note where those spots are so you can go back and sample from them. You only need about a 500-gram container, and you can sample from multiple spots and mix that together for one composite sample. But if you know that there's a yellowing spot, it's best just to take the sample from that yellow spot. The soil can be stored at room temperature or in the fridge. Those oospores, once produced, they don't really do anything, so storage isn't important. And then send or submit to your testing lab of choice. And really, it's important to check. If you decide where you want to send your sample, check their website, and they'll also have specific examples on how to send them the soil. I'm just going to go over quickly some examples of post-harvest fall soil sampling that we did a couple years ago in Saskatchewan. to show you that it, for the most part, it can work. And I have two fields that were kind of very different in presentation. And what we did here was we worked with producers to kind of identify a spot to sample from. That's here. They had said, oh, this was a low spot on their field. They thought it didn't do very well. And then we just took samples successively away from there to see how far away we could detect the Aphanomyces. And so in this case, and then we tested the samples, both the soil bait and qPCR method. So in this case here, this is our soil bait method. So it's our looking at disease severity. You know, that we were spot on with that spot number 6 being the place to sample that had Aphanomyces, and we could see it was quite high disease severity here. And all the rest were quite low and were usually more attributed to Fusarium than Aphanomyces. And then when we followed that up with our quantitative PCR, the results agreed in that we did test this as a positive sample, but we actually would have expected, based on the level of disease, that this should have been much higher. And then just to show you what it looks like from the soil bait assay, very, very clear-cut Aphanomyces symptoms. No doubt here that that was Aphanomyces. And then this is what samples look like from some of the other sites, where you can just see a little bit of Fusarium is starting there, which is why they have a low disease severity. And then we tested another field where this one had extensively yellowing throughout the field. So here we just basically started at, well, we still started at 6 and then moved out, but you can see that every single sample that we tested was positive. You do the soil bait assay again, very clear-cut results. Everything had very high disease levels. So we followed that up by testing it with qPCR. And we can see that the numbers are all over the place. They all did come back with a positive result, so that's good. But again, we would have expected these oospore numbers to be a little bit higher based on how high the disease severity was. And then just to show again how the soil bait test can be quite clear cut with that very clear Aphanomyces symptoms. And so we tested a number of fields. I think over 3 years, we ended up testing about 40 to 50 fields or so. And I just mapped them all out here. And really what happens is that the kind of lower risk fields that maybe have fewer, don't have yellowing all throughout, that just have some patches, we tend not to be able to detect those with qPCR, whereas the soil bait assay was telling that there was this disease throughout. And then the other thing you can notice here is disease severity of 5, which basically means the plant is dead. We see a huge range of the quantity of oospores. So there's still some challenges with this qPCR method, and I'm going to tell you why. I know I keep on telling you I'm going to tell you, but you will see it in a few minutes, what those challenges are. Okay, so we've kind of gone through 2020, you know, reflecting on your field, setting a baseline for future pulse crops in that field so that you can start making decisions. And so basically how to use this knowledge of soil testing is that if you had healthy pea and lentil fields and you did your soil test and it was negative, then I think you're good to go to plant peas or lentils into that field in 2024. If you saw a few yellowing spots and you did your soil test and you found that they were Aphanomyces positive, then it's definitely a time to pause and think about the next time you can plant peas or lentils into that field. And some of the information that I'll give you later on will help inform some of those decisions. If you saw widespread disease in 2020 and the soil was Aphanomyces positive, then it's a complete definite pause on putting pea and lentils into that field for at least 6 to 8 years, possibly more. And again, we'll talk about that a little bit. And the other important thing is to also think about the soil moisture patterns in your field in 2020. If you were in a region that was very, very wet in 2020, and to think about, you have to kind of monitor how that weather or what that soil moisture is going to look like for the next 3 years, because that can also affect those decisions in 2024. Okay, so now I'm going to move on to what you can do. So we kind of left 2020 behind. You've maybe taken some good records of how your pea and lentil fields did in 2020, done your fall soil testing, and you put that field aside until the next 3 to 4 years when you're going to plant a pea or lentil crop. So now you have to make the decision of what you want to plant and what field in 2021 and beyond. So how do we do that? And really, the most important thing that I can tell you is that it's all going to depend on your past field history, and we're going to go through that right now. Okay, so what we want to talk about then is soil sampling prior to planting peas or lentils. It would be great if when you're making that decision of where to plant your peas, you would know if Aphanomyces is present, and if it is present, at what levels. So I'm going to talk briefly about this idea of presence absence versus quantitative numbers. And I think what we're really trying to move towards is getting these quantitative numbers and being able to clearly define your risk. But we're not quite there yet. And that's because the threshold for detection is very similar to the threshold for quantification. So, you know, if you had a presence absence and it says, yes, you have it, then you know you're at risk. What we'd like to do, or what I'd like to do as the researcher, what I'm looking at, is being able to break that down into categories where we would say, okay, if you have 0 to 10 oospores per gram of soil, you're at no or a low risk. If you're between 10 to 100, you're at a moderate risk. But if you're at greater at 100, then you're at high risk. This is where I would love to move towards. We're not quite there yet, and I'm going to show you why. First, I think I've shown this slide in almost every presentation that I've given. I think it's the best slide to show you where the challenge with Aphanomyces is. 0, 1, 10, there's really no difference in disease. You hit 100 and then all of a sudden it's like you flipped a switch and you get a lot of disease. So it's like, how do we differentiate between 1, 10, and 100? And it turns out that that's very difficult to do. So right now it's, as I said, kind of presence is the same, our detection limit is the same as quantification. This is just showing the same thing on a graph where we see 0 to 10, very, very little disease. You hit 100, your disease starts to increase, and then past that, the disease severity doesn't really go up. And then we match that with DNA tests. And this is where we're kind of at right now. If we could accurately and reliably test the oospores that were in the soil, we would expect to see a curve like this. But when we do that for black soil, we actually fit fairly well onto the curve. For some reason, we get a few false positives lower on at the lower end of the scale. But here we can see we're pretty good. We have 10 oospores, we can measure 10 oospores. We move to salty loam soils, we can see that curve is starting to move below. Now we have 10 oospores, but we're measuring it a little bit lower here. And then we move to clay loam, and then we can see that curve is coming way below. So in this case, you know, we have 10, but we're pretty close, really not any significantly different than 0 here. We have 100 and it's telling us we have 10, and then we would think that that field was safe to plant in. So there's some challenges with this quantification of pathogens. And the reason for these challenges, I'm going to show this whole schematic again, like I promised I would. The reason for the challenges is this. When we're taking 500 grams of soil, we're starting with a fairly large amount of soil. Oospores are not uniformly distributed through the soil. They tend to clump because they're clumped on those pieces of decayed root. So we're taking our 250 milligrams of soil to put into our tube for testing. Chances could be that we didn't get a clump of oospores, that we just happened to get 1 oospore or maybe even 0 oospores. And so you move through that whole process and it tells you you have no DNA, and then you think you have a false negative, or you drastically underestimate the number of oospores in the soil. The other issue, and that's why I said these oospores are really the challenging beast here, is that under dry conditions, I'm hypothesizing or suspecting that the cell wall thickens during dormancy. And so maybe we did a good job of choosing our samples, but really an important step is being able to crack open these cells. And it seems like under dry conditions, we cannot get those cells to crack open. Again, we don't end up with any DNA to measure, and we get a false negative, or again, greatly underestimating what we would, what the true level is. And we've played with all sorts of ways to try to improve this cracking open of the cells. And so far, we just haven't found anything that worked. We are going to start looking at maybe trying to do a larger DNA extraction, but in terms of kind of a commercial test and the feasibility of extracting from 500 grams of soil, it might just never be cost efficient. So having said that, I guess I just wanted to kind of lay out what the cautions of soil testing is. Having said that, I still do recommend that you try to sample the fields of interest. And actually you can do, because now we're talking about fields that maybe had peas or lentils in them 3-4 years ago, you can do the sampling now in the fall, or you can do it in the spring. The ooze spore levels aren't really going to change that much. And so we do recommend this to try to give you an idea of the risk for that field. But having said that, after the whole spiel that I just went through, we do have to be aware of false negatives. So we're going to use additional factors to make the decision. And then also consider a soil bait test, I said you can do on your own. And so to maximize success of pre-plant sampling, it's very important that you choose your sampling spots wisely. Choose those low spots, those water tracks, seepage areas, low yielding areas. Also consider submitting multiple soil samples to test rather than just one. I know that there is cost involved, so it would be kind of the feasibility of that cost. But the more soil samples that you have, the more likelihood you are going to get at least one positive. And so aside from soil testing, the other thing that's most important is reviewing your past field history. The more times peas or lentils have been grown in that field, the more likely you're going to have high and optimum levels. The problem is that there's no magic number to say, okay, you've had peas in that field 3 times, you're done, you can't plant peas anymore. We don't know what that number is. Sweden, they did some research I think in the early 90s, and they found that some pea fields, if there was a pea every 6 years, some fields took 3 times of growing peas before the Aphanomyces problem got out of control. Other fields, it took 5 or 6 or more times before you had a problem. And the Netherlands, they had a similar kind of story. They did find that crop frequency explained some variation in disease severity, and that crop frequency was actually more important than that interval between crops. So this is the graph that I pulled from this paper in 1993, so it's not great graphics here, but really what they looked at is if you had 0 peas in 18 years, then your plants for peas would be healthy. If you had 1 pea in 18 years, you're going to start to see that you're starting to get some high levels of root rot up here. Again, 2 times peas in 18 years, your root rot levels increase, and then more than 3 times in 18 years, your root rot levels increase. And so then what I mean by saying crop frequency was more important than crop intervals, you know, if you did those 3 things of peas, 1, 2, 3 all in a row, and then didn't grow peas again for 17 years, you're kind of would be at the same risk as if you did your peas every 5 to 6 years. So kind of the frequency is maybe more important than interval length. Now the problem with this, like everything with this disease, is nothing is ever simple. It does seem to be that it is very field specific on how many times you can have a pea or lentil in that field before you're going to run into problems. And part of this is because there's so many differences in soil type, soil chemistry, you know, everything like that. And heavy textured, high clay fields do tend to do worse than kind of the lighter textured sandy fields that have good drainage. So that could be one of the reasons why one field it might have 3 or 4 pea crops, and then the other one you might be 6 or 7 and it does fine. Compacted fields also tend to do worse, so these are all important factors to take into account when you're reviewing your field history. A lot of researchers often just say there's other factors of which we are unaware. You know, we try to measure all these things, pH, different levels of chemicals in the soil, and none of them are really really good key indicators of why some fields do better than others. So in general, what we've kind of seen from that's happened in Alberta, Saskatchewan, and Manitoba, that it's about that 20-year history with a pea or lentil that you had it every 4 years. You know, gives you about 5 or 6 times that a pea or lentil has been in a field that you'll potentially start running into a problem. As I said, this is not a hard and fast rule, though. This is just very sort of general on observations. So when you're planning your seeding intentions for 2021, it's important to think back to how many times you've had pea and lentil crops in that field and how your pea and lentil crop did the last time it was in that field. So I'm going to go back to this, these same pictures that I showed before, except now we're thinking about what did your pea or lentil field look like 4 or 5 years ago. Again, if it looked like this, then you're probably okay to plant pea or lentils. I guess what there should say is that there's no guarantee that this won't be the year that you move from a field that looks like this to one that looks like this, where you start to see a few of these yellowing spots. So just keep that in mind. You know, if your field looked like this back in 2015 or 2016, then definitely don't plant peas or lentils in 2021. If your fields look like this, these again are the more challenging fields when we're trying to figure out what do we do with these yellowing patches. So for now, you know, what we recommend is soil test these low spots. This is where you want to target your pre-planting soil testing from these low spots, if you can remember where they were, or if you can remember the yellowing spots from 3 to 4 years ago. If not, then test the low spots and look at what that environment was the last time you grew pea and lentils in that field. And this is really important. So it's those wet years when you had pea and lentils that are really going to have the biggest impact on the next time you grow the crop. So a small patch, those small patches like I showed you in the previous slide, if you had really wet conditions, that can spread to the entire field by the next time you plant pea or lentils. For example, one of the sites where we do field trials, in 2011, this was a site near Lethbridge. They had some small yellow patches after extensive flooding. They didn't really think much of it. They thought it was more flooding issue. And then in 2016, the next time they planted peas, the entire field was infected. And then in 2020, where we now have our field trials, we almost got no yield from that field. So it just took that one kind of flooding event to move it from small yellow patches to an entire field. And 2011 and both 2016 were both very wet years, and 2020 actually. So we had 2011, 2016, 2020, all very wet years that had peas. And that really seems to be kind of the key recipe for these wrecks that we've seen. So when your fields that you're maybe planning for pea and lentil in 2021, think back to how wet it was in May and June, the last time you had peas or lentils in that field. And Sherry Lynn put together this very handy table that kind of summarizes moisture levels for Saskatchewan over the last 12 years or so. And so now if we're here into 2021 and you're thinking about, you know, planting your peas, fortunately we had some pretty intervening dry years back here. So if you're in 2021 and you want to go into a 2017 field, you should be okay because we had below average moisture in pea fields that year. If you're thinking of going into a 2016 field, then you might run into issues because for the most part, that was a very wet year. So again, this is just averaged across each location. You'll need to think about this specifically for your field. And I'm just going to, again, kind of show what this looks like from surveys where we really saw a big up and down in the prevalence and incidence of root rot depending on the years. 2015 was actually quite a dry year. And this is why we see very low incidence. But we went looking for fields with Aphanomyces so we could find them, but it actually wasn't widespread throughout the field. 2016, very wet year, we saw lots of Aphanomyces. 2017, 2018, it came down, didn't see very much. 2019 is a bit of an odd here because levels were quite high and the precipitation wasn't actually that high, but it's because there was some pockets that were really wet. And then the other thing is that if they're on fields, kind of rotation, just 2 years between rotation, you know, that were on fields that had peas in them in 2016 or even 2014 isn't shown here. It was a very wet year. That's why we'd start to see these numbers really go up. And then we see the same for lentils. I just don't have as many years of data as lentil, but again, wet years, lots of Aphanomyces, dry years, it does tend to go down. So the next question is, well, how long of an interval do we need between successive pea or lentil crops? Oospores are long-lived. They have a half-life of a year, and I'm going to go through kind of some hypothetical scenarios. And the easy answer is if your whole field is infected, don't even think about putting peas or lentils into that field for 6 to 8 years. Again, the exact interval time is unknown, and it will be field specific, and hopefully that's what our soil tests can help you with. So this was some research that was done in 2016 by Dr. Luke Baynard and Swift Current, and fields that we surveyed, he went back and tried to look at what the measure the Aphanomyces levels in the soil. And what he found was that on average, fields that had root rot and tested positive, on average there was about 1,000 oospores per gram of soil for pea fields, and surprisingly a little bit lower for lentil fields, on average about 100 oospores per gram of soil. So what I did was I just did a really easy calculation of looking at if we cut that number in half every year, how many years it would take. So if you're sitting at about 100 oospores per gram of soil and you start decreasing that amount every year, you're kind of at that safe level below 10 in about 4 to 5 years. If you're at 1,000, you can see that it takes you kind of 6 to 8 years to get below that level. That's kind of where that 6 to 8 year number comes from. If you're really high, and we did see a few fields that had these high 10,000 oospores per gram, then you can see that even after 10 years, we haven't hit that. So this is why I think the research is so important, that we are able to accurately quantify oospores from soil. Getting short on time, so I will try to run through this quickly. Just some examples of what we've been looking at in terms of this crop interval. And again, I'm trying to highlight that it's very field specific. One of our field trial sites is in Red Deer. This is in the black soil zone. Great environment for growing peas. And what we can see with the last pea crop in 2011, by 2018, we saw a really nice reduction in the root rot levels. And we saw the yields kind of come back up to what can be kind of normal in that area. And we'll see that there was some intervening dry years there. So those dry years really helped to kind of bring down the inoculum, but it still took from 2011 to 2018. So a break of 7 years, 6-7 years to come down. Our site in Tabor, which is very close to the Lethbridge Research Center, the last time it had grown peas was in 2014. Again, despite these intervening dry years, we really haven't seen the disease severity come down. On the yields, we haven't really seen much change in yield response. We didn't have a trial there in 2016, that's why there's no data, but it actually would have been a very wet year. So again, just kind of showing, this one's not as long of a difference. It's only 5 years in between. So who knows what will happen this year. Actually, I have data for that in the next slide, and we will see what happens there. And so the other thing that we're doing is this ongoing rotation study, where we are actually, rather than just planting peas in a new spot every year, this one's actually part of a study where we're changing the frequency of peas within a rotation. We're into year 3 of a 5-year study. We have a location here in Lethbridge. So I'll just walk you through what this means. So the last time they had peas was in 2016, and then one of our treatments is peas every other year. So we had peas in 2016, we had peas in 2018, and then this is showing what the disease severity looked like for peas in 2020. And we could see very high. So pea every other year, very high disease. Surprisingly, when we're looking at our peas, our 3-year break between peas here, I did not expect this number to come down this much from 2016 to 2020, but it did. Oh, just to show every field is different, and you never know what you're going to get. Again, lots of dry years in there, though. And then we're also just testing alternate pulse crops, and this is soybean. We can see very low disease. This is our Tabor site, the same one that I was talking about previous. So we had peas in 2014. Then we started the trial in 2018, so that's why it was peas again in 2018, peas in 2020, very high. And now we can see, the disease severity is coming down relative to this one, but it's still quite high. So why such a difference in these two fields? We don't know that. I think that's one of the still very big unknowns. What is that specific field conditions that is driving these differences in how disease will present? So we are looking, there's a lot of research looking at this agronomics and looking at methods to try to reduce the ooze spore load in the soil and kind of these intervening times between pulse crops. We're supposed to start a big project on looking at the rate of ooze spore decay under prairie conditions, so we can determine if that half-life of a year was correct, or, you know, with our more extreme prairie conditions of really hot, cold soils, really wet, dry soils, how that affects ooze spore decay, whether there are some rotational crops that are maybe better in a rotation than others to help reducing ooze spore loads, looking at incorporation of green manures or lime products. We continue to evaluate seed or foliar treatments. It's just really challenging, you guys, and like no matter what we try, so far we really haven't had great success. There's no silver bullet for this disease. And something that I'm starting to think more about is patch management. And I think this is going to become very important moving forward, where you have those yellowing patches in your field, and we can tell you what to do with them to try to reduce spread throughout the whole field. But we're not there yet, but that's my dream, is that we'll be able to come up with something. But the foundation for all of this is accurately quantifying ooze spores in the soil. I'm going to talk very quickly. I want to leave time for questions, but talk very quickly about interactions between Fusarium and Aphanomyces. I think you've all heard this before. Basically, Fusarium just makes it all worse, and so we are targeting some research as well, trying to reduce Fusarium. And if you go to, again, one of the back sheets on the Sask Pulse Growers, there's a very nice description of Fusarium root rot and pulse crops. So I'll direct you towards that rather than getting into too much into Fusarium. So reminders for 2021, choose your fields for pea and lentil carefully. Review the frequency that you've had pea or lentils in that fields. Think about how your pea and lentil crops did in those fields, whether there was very wet conditions in those fields the last time pea and lentils were grown, and just what the conditions of that field are in terms of soil. Is it very heavy clay soil? Is there issues with compaction? On top of that, do a soil test. I think heavily recommend this, but just be cautious about those negative results. Don't think that it gives you a free pass to plant Aphanomyces in that soil if you've checked off some of these boxes for having high risk. Consider using seed treatments, especially for the low risk fields or the patchy fields. You know, at this point, I would recommend, because 2020 was a wet, I guess it's, sorry, for 2021, it depends on how your field did 3 to 4 years ago. But, you know, patchy fields, I wish I had a concrete answer to be like, no, don't plant in a patchy field. Yes, plant in a patchy field. I would say err on the side of caution and try to stay away from those patchy fields, give them a little bit of a longer crop interval, and really try to focus on the results of those soil tests. And the other thing that I want to stress for 2021, and that I think should become habit for everyone growing peas and lentils, is to check and test your roots. That gives you the best information. So management decisions for 2021, just as a reminder, there are no in-crop options. So once you've committed to growing peas and lentils into that field, you have to make all your decisions prior to planting. And some of those decisions include choosing good quality seed, using seed treatments to reduce early season stresses due to seed-borne pathogens. So, you know, to reduce Fusarium, reduce Pythium, Rhizoctonia, all those other players that are part of the root rot complex. Choose varieties that are well adapted to your growing region by consulting regional crop variety guides, and that maybe have some tolerance to Fusarium species, although that is still a little bit unknown how that might play into it. You know, and everything else agronomically that you can do for that crop, your optimal nutrient levels. If you're in an area that has a pea leaf weevil risk, use a seed treatment, because just because it always seem to just have like the worst news. There is some interaction between pea leaf weevil and root rot that can make root rot worse. And then minimize practices that will lead to compaction, such as don't roll under wet conditions. And finally, I said I was going to end my presentation with this, and I'm going to, because I think it's really the most important thing to do is check and test your roots. Shoot symptoms are sometimes not obvious, even if there's root rot, and I have lots of pictures to show that. Here, 2 plants growing side by side. These ones are yellow, these ones nice green healthy shoots. Look at the roots, they both have root rot. Again, here's some pea plants earlier on that actually looked like they were quite healthy. You pull them out and you can see that the root rot is there. And then the same with lentils, and these you can see that there's some yellowing, but there was some green patches in there too. So it's very important to check your roots. And all the issues with the false negatives for soil testing, root testing for DNA and quantification works remarkably well. You don't have to worry about false negatives. Now we're in the lab, we collect thousands and thousands of roots, we take them home and wash them. I know that's not convenient to do. Do you have to wash your roots to see symptoms? For the most part, you don't. You can just pull them up in the field if it's dry and you can see nice symptoms. Here's an example, maybe when we didn't. We pulled these out, see lots of soil attached to the roots. We actually thought they're healthy. We washed them up and then you can see that there was root rot starting. But here, another case, we pulled these roots straight up from the soil. You can see the symptoms of Aphanomyces right away without having to wash them. If you want to see kind of the distinguishing between Aphanomyces and Fusarium, then you usually have to wash them to start seeing some of that blackening and other issues. But then if your root rots are really bad, you often don't have to wash them at all. You can just pull them out of the soil and tell right away that you have no roots left. And then I'm just going to end here with, I guess, what my dream is for managing root rots, or if it was in a perfect world, and these oospores weren't so challenging to work with. What I would always recommend to start with is a soil test. Before any decisions for pea or lentil planting, test your soil. What that is dependent on, though, is reliable oospore quantification. And like I said, we are trying to work on that very, very heavily. It's just proving to be challenging. And moving towards this idea of patch management. Once you get your soil test results, then if we can build these oospore decay models where we can determine, okay, what's your starting level, how many years do you need until you can plant pure lentils, you'd be able to know what that interval period that is needed. And then you could plant your partially resistant cultivars and agronomics, which are also in the works here. You know, we have a good base then for being able to plant in these fields where we know what our risk is. And for that, we do have to know what the acceptable oospore level is. We're not going to be able to plant partially resistant cultivars in these heavily infested fields. It will lose the resistance it has too quickly. So this is all dependent, on each other. And with that, I'll end anyway to say thanks to my team. This was last Christmas when we were allowed to actually see each other. Now we just see each other over Zoom. And then finally, thanks to funders and all the collaborators that work very hard on this research. And with that, I ended up going a little long. Hopefully there's enough time for questions.
Well, thank you, Dr. Chatterton. That was awesome to see the presentation and the information that you're providing. It looks like it is going to be great information for growers and agronomists to use for predicting risk going into 2021. We're now ready to entertain some questions, and the questions were coming in like crazy, so we'll try to do our best to get through most of them. I've tried to group them together so that they're kind of all on one subject matter at one time. So even though your question may not be worded exactly how you brought it in, it hopefully will give you the answers. Just again, this webinar is being recorded, so you will have the ability to go back. The questions will be part of the recording as well. So the first question, I'm going to focus on questions that were related to kind of the lab analysis and the soil testing. And just give Syama a couple seconds to catch her breath as she presented so much information. The question that we're going to ask first revolves around testing going into a pea and lentil crop for 2021. And the big question is, are you recommending soil testing for fields prior to planting in 2021? Is there quantitative tests right now that are accurate enough to identify fields that are better candidates? And where do they find out about lab capacity? Some of the land might be new and cropping history isn't available, and so growers and agronomists are wanting to soil test to see where the levels are at right now.
Yes, really good question. I am recommending soil testing going into 2021. Because even if the quantitative part isn't there yet or isn't completely accurate, even a presence absence is a good starting point. So I would recommend it. Now, if you're going into a field that has never had pea or lentils before, the likelihood that there will be high levels of Aphanomyces are quite low. And I know it comes down to this question of what's a true negative versus what's a false negative. So you do have to take those soil testing results in conjunction with the other things that I mentioned. So if you don't have any crop history, there's never been a history of pea or lentils in there. The last time you grew pea and lentils, they did really well, and your test comes back negative, then that's a pretty good indicator that that's a true negative. If you did a test and it came back negative, but you knew the last time you grew peas or lentils in there, it was a wreck, and all the other, you can click all the other check marks, then I would be cautious about that result, and then maybe try to follow up with soil-based tests that you can even do on your own.
Okay, and that answers kind of the next question is, can a soil-based test be done prior to planting in 2021? And obviously the answer is yes. Absolutely. Where would growers and agronomists go to find out lab capacity and what labs can do what test?
That's a good question. And I myself don't know what method every lab uses. Sherilyn, you might have had, can't remember if you had some information on that, or you might just have to go to each lab's website and look at what their information is on how they do their test.
Okay, we did, just so the audience knows, we do have a fact sheet that was just updated this fall on testing for Aphanomyces, and it lists all the labs across Canada that do testing that they can send soil or plant material. Again, it's always good to just check with the lab to make sure that the procedures haven't changed, but there is that available, and we will include a link to that fact sheet as well after the webinar and when we send out the links to the other resources as well. Another question is, we have found variability in soil testing results and not getting any clear explanation why they're seeing the variance. Samples were submitted in the spring and we're coming back at 1,300 spores per gram of soil. Then the fall soil sample was submitted and came back at 0. What effects does drought have on the sampling procedure or depth, and how do we utilize the test to make better informed rotation decisions?
Yeah, and that is a very good question, and definitely one of the biggest challenges that I am working with in terms of research, because we found the exact same thing. Our fields that we do our field trials in, that we know have high Aphanomyces levels, when we tested them under drought conditions, they come back with 0 oospores, and we know that that is incorrect. And so I think that is the #1 reason, is these drought conditions. And I suspect, it's like I showed in one of those slides there, it's the ability to break open those cell walls of the oospores after they have gone into what I call super hardcore dormancy. It's not a very scientific term, don't quote me on that one, but I suspect that's what's happening. The cell walls are becoming thicker, they're becoming more resistant to kind of environmental degradation, and then we just can't crack them open. If we can't crack them open, we can't test the DNA. So we are playing around with some things of looking if we can re-wet the soil, let it sit, if that'll help. But it might just be going back to a soil-based test. And again, it's kind of using, I guess, some intuition or common sense that you're like, okay, I know these results aren't correct based on everything else. I know that's a challenging answer because I'm preaching soil testing. And I think it's important to get into the habit of soil testing because, like I said, it's going to be the foundation for management. And hopefully we will come up with some new methods. And we are looking at doing some other methods of researching some other methods of soil testing, as we speak. But, yeah.
Okay, thank you. So continuing on with the soil testing, when doing multiple samples, how far apart should they be in a field, and how many sites per field should be sampled?
Yeah, so when we did kind of the fall testing that we did a few years in a row, we were going about 100 meters between samples, and we did 10 samples per field. And we found with that, we could generally pick out one positive site out of the 10. I know paying for testing of 10 soil samples is a lot though, so that might not be feasible. But I guess that would be, another recommendation. If you do a soil test and it comes back negative and you suspect you have Aphanomyces, try again in another location. I don't know if that totally answers the question, but I'd say, yeah, 10 if you can afford it.
Okay. Now, and then the last sort of lab-based question is, has anyone reached out to the test labs in Western Canada to see what percentage of samples they're getting and what the results are showing? Kind of a survey of the labs. Has anyone done that?
I don't think so. And I guess would be the question whether they'd be willing to share that data with us or not.
Yeah, okay, something we can follow up on.
Yeah.
So we're going to switch gears here a little bit because we've got lots of questions, but I'm trying to kind of cover the different sort of areas of questions. So one is on the environment here. How possibly did the rapid dry down of the fields in central Saskatchewan that occurred more or less in the whole month of August affect the Aphanomyces zoospores' ability to multiply? Can we anticipate lower numbers? There was fields that had 80% damage of Aphanomyces, but they still kind of came back and produced 50 bushel pea crops. So I guess a comment on, when the conditions dry up after you do see the infections, can you expect lower spore loads at the end?
I think unfortunately not, because what will have happened is that you had your infection when it was wet, and then as soon as those conditions became unfavorable for the pathogen to remain infecting and colonizing the plants, they would undergo fertilization and produce oospores. So you're probably likely to have just as many oospores there now, even though it was dry. So I would still consider those fields as being a high-risk field if you saw that level of kind of infection early on in the season.
Okay. Next question, is there a time during plant life when the disease will not affect the plant? An example of getting moisture when the pods are filling after it's been dry for most of the growing season?
Yes, absolutely. So the earlier we see infection, the bigger impact it's going to have. And that's why it's the May and June rainfalls that really cause the most damage. If you get rain kind of into July during pod fill, you're really not going to see very much damage because those roots, first, they're very well developed, and so they can sustain a little more infection, and then you just don't have the length of time to kind of get through a whole disease cycle.
Okay, thank you. The next set of questions relate more to host crops and rotational crops. And just if you can kind of go through the information pertaining to what crops, pulse crops, Aphanomyces infect. And the question pertains to, can chickpeas, soybeans, or faba beans be used in rotation with peas and lentils to allow you to extend those rotational crops?
Yes, absolutely you can. And I was trying to fit that into my presentation, but I didn't have time. I think it's from the, because in the webinar I gave in March, I talked about that. So yes, chickpea, soybean, faba bean are all good options if you have an Aphanomyces infested field. There are some varieties of faba bean that do appear to be susceptible, but those are the older varieties. So the newer ones, the newer small seeded varieties seem to be resistant. The same with soybean. Chickpea supports a small amount of Aphanomyces growth, but really not a lot. And we don't actually see it causing a lot of damage to the roots, but we can find oospores in the roots every once in a while. And it may be worth looking at that, you know, if chickpeas could actually act as a trap crop where they're inducing those oospores to germinate, but the oospores can't multiply and reproduce, or reproduce and then multiply and get left behind in the soil. We're looking at that with including chickpea in a rotation. But yeah.
Okay. And then what about alfalfa? Is that a concern in, you know, a long-term alfalfa field and going into peas and lentils?
Yes, tough questions. Alfalfa is a tricky one because it is a host of Aphanomyces, but Aphanomyces has race structures on alfalfa. I think there's a race 1, a race 2, and a race, I think they just found a race 3 in the US. I don't have the slide in front of me and I don't know if I can remember it offhand. So I think race 1 can infect peas and alfalfa, but race 2 can't infect peas. It can only infect alfalfa, or could be vice versa. And we don't necessarily know what the race structure is that we have because you can't tell from testing it against peas. You have to test it against alfalfa. And then the other issue is that in Canada, we don't seem to label alfalfa varieties with the type of resistance that they have to Aphanomyces. So you might not know, so you wouldn't be able to tell what alfalfa variety, what type of resistance it had anyway. You know, maybe as we're seeing Aphanomyces becoming more challenging, and if it does start to become an issue on alfalfa, then we would have to start discussing moving towards labeling of alfalfa varieties.
Thank you. Would non-susceptible cover crops reduce oospore populations due to better water infiltration rates? And would legume cover crops have the opposite effect?
Yes, that's something that we are trying to look at right now. In terms of legume cover crops, some of them are okay. Stay away from vetches. Vetches are maybe more susceptible to Aphanomyces than pea. And I know it's, you know, sometimes vetches can occur as weeds too, so that could be an issue in the field. But other legumes like clovers, lupins, try to remember all the ones we've tested, do seem to be fairly resistant. And I think in the webinar that I gave in March, I did list what all those different legume crops were and their susceptibility to Aphanomyces. Now, in terms of other cover crops, yes, we are looking into that. Things like rye, oats, brassica, cover crops, both for their impact on, you know, reducing soil saturation, moisture effects, and also the idea that they might have some biofumigant properties themselves that will help to speed up the decay of oospores. Unfortunately, it's been a bit of a frustrating trial, and we just don't have data on that yet.
Okay, so you kind of mentioned the biofumigant properties, and there was a multitude of questions that came in on including other crops in the rotation that could potentially act as biofumigants or oospore germination stimulators to try and draw down the spore load. There was a mention and question about canola, mustard, oats, durum. Do you have any comments on that? I know you're doing some work right now, but maybe a little bit on sort of the theoretical background on that.
Yeah, so some of the background or some previous research does show that mustards, and particularly mustards that have these high glucosinolate levels, which I don't know how you would know exactly what mustards have high glucosinolates, but mustards with these high glucosinolates, which is what acts as the biofumigant, and oats, there's been research done that shows that those can help reduce oospore levels in the soil. But a lot of that research was done in greenhouse trials and never brought out to the field. So it is what we're trying to look at it now as field trials. Something like canola, unfortunately, I mean, it's the most common, I call it the common rotational crop, but I know it's usually the other way around. It doesn't have any glucosinolates, so you're not going to get the biofumigant effect from canola. And certainly with just how commonly canola is grown. You know, if it has an effect, we would maybe see that in the pea fields as it is. So mustards and oats seem to be the ones that will have the greatest potential for that. And those, we are specifically looking at those two types of crops for their ability to do that.
So would it be, would it be safe to say that kind of the more crops you can include in the rotation to spread out the rotation outside of the biofumigant activities is a good way to also start looking at rotations, increasing the diversity, I guess?
Yes, absolutely. Increasing the diversity is always good. You know, just for the soil health. And I think that's when I said kind of unknown factors that we're unaware of, this idea of soil health and how soil health plays into a field being able to support lower population levels or higher or lower pathogen levels. You know, soil health is probably a big part of that, and we just haven't figured out what all those different aspects of soil health are yet. But definitely crop diversity improves soil health, and that's always a good thing.
Okay. So the last kind of section of questions that came in are related to sort of the spread of the oospores and the pathogen within the field. So any comments on the tillage and the effects of movement of Aphanomyces in the field? Does it spread similar to clubroot? Should there be sanitation between fields or other products that should be used to sanitize equipment?
Yes, exactly the same way as clubroot. So the only way that it can spread is the oospores in the soil. So anything that moves soil around is going to help move, you know, the inoculum around within your field, and then also from field to field. So there's been a lot of information on sanitation for clubroot, and I would say follow the same practices for Aphanomyces to reduce moving it from field to field.
Okay, and then the last question related to that is with clubroot, we know it kind of started in one central area and then has been spreading outward from that, where Aphanomyces, we seem to have it across Western Canada. Is there any differences in terms of how it's spreading, or if it's if the Aphanomyces is more of a pathogen that's just present in low amounts kind of everywhere, or if we are actually moving it from field to field?
Yes, another good question. The hypothesis is that it's a native pathogen that's just in low levels everywhere, and this is why crop history or crop frequency is so important, because every time you're putting a pea or lentil field into that pea or lentil crop into that field, you're basically bumping up the Aphanomyces levels just a little bit, little increments until you hit that threshold level. Again, there's probably some unknown factor having to do with fields on that, because you know, some people will have grown peas and lentils for a long time and never have a problem, and other people it's 5 or 6 times and there's a problem, right? So exactly what that is, we don't know, but yes, the hypothesis is that it is just present in all the soils across the prairies.
Okay. So I think with that, we're kind of, you know, went past what we were targeting, but there were so many good questions that came in, and tried to answer as many of those as possible. I have one final question. I'm hoping it's an easy one for you. So as we're getting to the end of this and also because we're in the fall situation right now where our soils are really cold and likely even frozen in some cases, is can you get a reliable soil test from frozen soil samples?
Yes. It is actually a difficult question,
Oh.
but you still can. Yeah, it's like, I'd have to think about that. Yeah, you still can get a reliable test from frozen soil samples. And the reason why I say that is because when we did that big project where we go collecting all those fall soil samples, we actually collected them, tested them, froze them, and then went back and tested them like 3 to 4 years later, and we still got nice results. So a frozen soil sample will still give you good results.
Awesome. Thank you so much, Dr. Chatterton, for your presentation, as well as for answering the multitude of questions that came in. Reminder for those that are attending that your CCA numbers, for those that will be wanting credits, an email will be sent out shortly, and then you can provide your CCA number. At that time, you can also provide suggestions for future webinar topics, and feedback and suggestions are always appreciated. This webinar was recorded and will be posted on our saskpulse.com website, as well as our YouTube page, and it will also be shared on our Facebook and Twitter handles. For those attending, we will also be sending out an email that has a link to the recording, as well as the fact sheets that we mentioned within this presentation. Thank you again to Dr. Chatterton for providing the listeners on how we can reduce the risk for root rots in peas and lentils before we put seed in the ground for 2021. As well, a big thank you to all the participants for joining us today. With that, everyone, have a great day.